to have atherosclerotic lesions, and that's well understood now from some of the pathology data that came after the Vietnam War. There's a lot of difficulty because you often compare young cases with premature coronary disease with aged controls. At least until recently, invasive screening of the coronaries was not possible, and surrogates had to be used, such as looking at the carotid intima thickness, looking at other measures of endothelial dysfunction. But more recently, with CT scanning, coronary calcium scoring, it's able to look at these so-called intermediate phenotypes. The simple matter of getting a clear family history is very difficult, and there's the issue of survivor bias. If you've died of the disease, you're not going to be around to be included in the studies. There have been legions of candidate gene association studies, whether they be looking at genetic impact on lipid metabolism, on thrombosis, on platelet function, on issues of blood pressure regulation, on insulin metabolism, homocysteine, or on the inflammatory response. These studies will build on our existing knowledge by investigating the effect and the variation in genes on this pathophysiology, but it's a complex process. These studies are very powerful, and they're often informed by other data, particularly basic cellular work, animal studies, so that if you find a true association, it's likely to be a true positive. And the effects of these gene association studies can be amplified when you look at gene-environment interactions. Many studies have been done looking at the ACE gene and left ventricular hypertrophy and the effects of exercise. But these sort of studies need big samples, and they need some sort of starting building block of knowledge. And furthermore, because you're picking and choosing, they emit many genes and gene variants you may simply not be aware of at the start. So a certain amount of assumed knowledge is required. The risk associated with these single nucleotide polymorphisms, or SNPs, may only be quite modest. There's always the problem about chance associations coming up, the difficulty in reproducing findings in serial studies, the fact that we often underestimate the true frequency of the SNPs in the control population weakens any association. There's often poor matching for ethnicity, badly powered studies with inadequate statistical power, and there's publication bias. You know, if it's a negative study, nobody wants to read about it, even though the findings may be true and valid. So gene association studies and a candidate gene approach is very much like the bedraggled England supporter with his empty bottle of Jack Daniels looking for his keys in the dark. Now, things have moved quite a long way from the original paper of Watson and Crick, if you can read at the back, was 1953, with the completion of the Human Genome Project at several $1 billion cost. We now know that there's a catalogue of about 3 billion DNA base pairs that make up all 46 of our chromosomes, and we can identify the products of the 100 or so human genes, and this has taken things forwards in huge leaps and bounds. At this point, it's probably worth just spending a couple of minutes on some basic genetic terminology. As I briefly mentioned before, the terminology of a single nucleotide polymorphism, or SNP. A SNP really is a DNA sequence where you have a single base pair variation where one person might be AAGCCTA and a single base pair changed such that you have one allele with a C and one allele with a T. One of them will be associated with risk, hence a risk allele, and one is so-called minor allele. And we know that SNPs are quite common and that they occur about 1 in every 300 base pairs along the genome. Furthermore, we have the terminology of something called a haplotype, which is the specific combination of alleles at multiple different loci or positions that seem to be transmitted together on the same chromosome. Linkage that's very difficult with a dry mouth. Linkage disequilibrium, that's why I went to intervention, it's much easier, is the non-random association of alleles at two more positions, two more loci, they're not necessarily on the same chromosome. And in this way, we can look at how blocks of genetic information are inherited together. And this issue of LD linkage disequilibrium describes a situation where combinations of alleles seem to occur more or less frequency in your population of interest than would be expected through random association alone. In this way, it's possible to identify the commonalities in many parts of the chromosome, such that only a relatively small number of haplotypes are required to identify a large percentage of the population. So in a given population, about half may have one haplotype, about a third may have a second, and about 8% may have a third. So looking at 3 blocks of genetic information gives you a lot of information about the whole genome. In this way, it's possible to build up so-called haplotype maps, where one SNP can act as a proxy for many. So if you look at 4 different chromosomes, you've got the bases running along, there'll be SNPs at 3 different positions, and these SNPs are associated as a haplotype and inherited together. And these in turn can be identified by so-called tag SNPs. So you can use very simple genetic techniques to identify very subtle variation, often many tens of thousand bases apart, often on different chromosomes, by merit of the fact that these SNPs are inherited as blocks. So only a relatively small number of genotypes will provide information over a very large area of the genome. Now, with the completion of the Human Genome project, and subsequent to that, the HapMap project, where the patterns of LD have been identified, there's now the next breakthrough, and this is where we are in the, sort of the current century, of performing genome-wide association studies. Now, genome-wide association studies look for associations between single genes in an unbiased fashion with no predisposition or early idea of what you're looking for. And even though there are about 7 million common SNPs, if you take a minor allele frequency of about 5% in the human genome, we know that because of identifying the linkage disequilibrium, it's possible to identify the presence or absence of these associations relatively easy. And by taking advantage of these patterns of association, if you genotype about 500,000 base pairs for the SNPs present, you can cover about 90% of the human genome, rather than going through all 7 million of them. The problem is that it creates a lot of statistical difficulty, and you have the conundrum of multiple comparisons, and you have to do a Bonferroni correction, and the statistical power required of any association has to be much, much higher than the normal less than p equals 0.05, as is present in many clinical studies. So essentially, the basis of genome-wide association studies is to take a large patient sample, you have your control sample, you genotype them usually using these computer chips, which look at about 500,000 different SNPs. You look for any chance associations at very high statistical level, and then what you have to do is repeat the whole process in 1, 2, 3, 4, 5 different genome-wide studies. And if the statistical association still holds, then it is very likely that the observation of a particular defect in a particular condition is true rather than by chance. So the question, is there a gene for coronary disease? Well, possibly. You won't get a straight answer, I'm afraid. When I was asked to talk about 3 years ago, it was literally about 2 weeks after a series of 3 papers were published in Science. And this seemed to allude to chromosome 9 as being where the money was. And the first study by McPherson's group looked at using chips of about 100,000 SNPs, and they analysed 3 sequential case control studies, the first 2 Ottawa Heart studies and then the ARIC study. Now in the first study, it's again case control, they identified about 2,500 SNPs that seemed to be statistically associated with premature coronary disease. When these same SNPs were looked at in the second Ottawa study, about 50 were still apparently associated with coronary disease. And then when they put it through the ARIC database, 2 seemed to remain. And these were both located within about 58,000 base pairs on the short arm, that's 9p21 of chromosome 9. So this is certainly an area of interest. In that same Science journal, this finding was validated by a separate series of studies where they then went on to look at 3 further cohorts. There was the Copenhagen City study, the Dallas Heart study, and the third Ottawa Heart study. And this is interesting because the 2 alleles that they identified on 9p21 seem to define a 15 or 20% increase in the risk of coronary disease in the 50% of the population who carried 1 abnormal copy, and a risk of about 30-40% increase in the homozygotes who had the two copies. And it's important because these gene defects are common. And the associated population attributable risk with these alleles was about 12 to 15% in the ARIC study, about 10 to 13% in the Copenhagen group. And this is also important because about 1 in 3 people in these studies who were identified as having premature coronary disease seem to carry 2 copies of the abnormal gene, compared to about 24% of healthy people. The corroborative evidence was produced by an Icelandic group, And in exactly the same way, they did a big case control study of about 8,000 patients and had the same sort of finding. This was then confirmed in another serial analysis of over 4,000 cases and controls from Philadelphia, Atlanta and Durham, totaling about 16,000 patients. So there's a big population studies. And 21% of the population were homozygous for the variants, and that was associated with a 60% increase in premature coronary disease, particularly of myocardial infarction. And in those people who had in fact come through with early myocardial infarction and survived, so there's this issue of survivor bias, there was a twofold increase in risk in those homozygotes. And they felt that the population attributable risk of myocardial infarction was between 20 and 30%, so big numbers. And this was finally confirmed by the third in the three publications by Professor Samani's group. And they looked at the Wellcome Trust Case Consortium and the German MI family study. The technology had moved on. They were able to use these gene chips looking at 350,000 SNPs. And they did this in the 5,000 cases and controls of the Wellcome study. And it replicated the same sort of findings also in the German family MI study. And of the 400 genetic abnormalities or variants that were found in the Wellcome group at a statistical level of p less than 0.05, only 3 still held true when they looked at the German MI study. This was 9p21, a variant on the long arm of chromosome 6 and the long arm of chromosome 2. The individuals who had these SNPs seemed to have between a 1.2 and a 1.4 times risk of myocardial infarction. These were common alleles present between 25 and 50% of the population, so that the population attributable risk is quite significant. Since 2007, and you're not meant to be able to read this, there have been more than 40 studies looking at this area, 9p21, so there's obviously something in it. The levels of statistical significance are high throughout by definition. They have to be for genome-wide studies, so this is more than just a chance finding. If you look at people who had 2 of the at-risk alleles, comparing those to 1 at-risk allele, the overall odds ratio is about 1.25, significant increase in the risk of myocardial infarction. And interestingly, it seems to have a greater effect in people who have premature coronary disease. The earlier the infarct, the index event, the greater the risk. Similarly, the other side of the equation is true. If you look at individuals who have no at-risk alleles and compare those to the 50% of the population with 1 at-risk allele, there's about a 20% reduction in the overall risk of myocardial infarction. So what does this area on 9p21 actually do? Well, it's interesting because this area seems to code sequences for the cyclin-dependent kinase inhibitors. Nobody's quite sure exactly what these 2 enzymes do, but they seem to have a role in the cell cycle. They've been thought to have an impact on tumour turnover, so whether that has something to do with how plaque burden changes, nobody really knows. And the same area of interest on 9p21 seems to be associated with a susceptibility to generating diabetes. So it's possible there's a shared mechanism. It's not just 9p21, there are about 11 likely candidate genes that have been flagged up. But the interesting thing about them is that a lot of these variants are common. In chromosome 1, that's present in about 77% of Europeans. In chromosome 6, again, similar frequency of the risk allele present in 78%. But the thing about this is nobody quite knows the underlying mechanism. A lot of these variants are not associated in coding areas. So there's a lot that we don't know. So I suppose the summary statement with respect to what we can say about the novel loci and their association with risk of myocardial infarction and coronary disease is that the strongest and most replicated effect is with respect to variants at 9p21. Only 2 of the 11 chromosomal regions identified appear to affect traditional risk factors. The risks alleles that we have identified are very common, such that 50% of Europeans carry 1 abnormal copy of the 9p21 risk allele, and a quarter will carry 2. That means a quarter of you are probably okay. Each risk allele seems to increase the risk of myocardial infarction by between 10 and 30%. If you're a homozygote, that's about a 60% risk increase, compared to the 25% who do not carry any of the variants. But importantly, the high risk of the alleles explains why the population effect is very great, because even though the effect is small, the penetrance is high. And the genetic epidemiological data demonstrate that all of us carry a very